Okay, I think we'll get started. We've got a bunch of people showing up, so keep coming. Thank you, everybody, for coming to the seminar today. I'm super excited to introduce Dr. Verzi. Dr. Verzi received his PhD at the University of California in San Francisco in molecular biology, and followed that up with a postdoctoral fellowship at the Dana-Farber Cancer Institute at Harvard. He was also an instructor and then was recruited to Rutgers, where he's been progressing through the ranks as currently an associate professor in the Department of Genetics. He has a litany of awards, R01s, co-PI, U01s, phenomenally productive, high-impact papers, and an excellent colleague and speaker. I've had a chance to interact with Mike on the DDKC study section, where his comments are always very insightful and helpful to the entire group. Mike shares my interest in trying to understand a regulation in epithelial cells within the gut, and really uses a sort of genomics approach to narrow down into fundamental mechanisms that drive physiological pathways of key significance in the intestine. So without further ado, Mike, welcome, and thank you for speaking at our seminar. We're looking forward to learning more about your work.
Thanks, Shazad. It's really great to be here virtually, and I see a lot of familiar names in the Zoom windows here. So I'm excited to be with you guys virtually again, and it's been fun to serve with you on this committee as well. I have to point out that Shazad, your passion for mentoring and supporting the trainees that apply to the study section we serve on it really comes through during your reviews. And I didn't get a chance to mention that to you earlier when we were chatting. And I know John's on, he's a great mentor as well. And it's really great program that you have there together at UNC Duke. It was great to chat with Scott earlier, and look forward to interacting with you guys more in the future. So today I have about 40-minute presentation, give or take, that I'd encourage you to unmute and interrupt and ask questions if you have some along the way, because I built in some times for questions, and I know the Zoom meetings, you don't want those to go on too long, so I thought it'd be a little more fun to have it be a little bit interactive. So today I'm going to tell you a little bit of effort we've put together recently to try to pick about how this really interesting structure at the apical surface of the enterocytes is built. And that's the structure known as the brush border, and particularly a body of work where we've shown that the HNF4 transcription factor coordinates a large or the majority, we think, of the gene expression required to assemble a brush border. We found that the brush border gene program under a control of HNF4 isn't unique to the intestine, but actually a very similar program helps the kidney proximal tubule cells make brush border, and reproductive tissue, epithelial in these tissues, make a brush border as well. I'll talk a little bit about future directions in our efforts to understand brush border gene regulation in some disease contexts, for example, to maintain the stoichiometry of the proteins that localize to the brush border, and then briefly share some other ongoings in our lab, maybe in hopes of triggering some ideas or some collaborations with you all. So I'll begin with introducing the brush border. So the intestine, especially the proximal intestine, is all about surface area to absorb nutrients. Form follows function. We all learned that in middle school biology, probably. So this is the duodenal villi shown by SEM, or in a H&E through a villus. You see a lot of enterocytes here interrupted by a goblet cell, where there's kind of a thin red line there, and then overlying that is the brush border. If you zoom in on TEM, you can see that making up this structure are these microvillus projections. They're about 2 microns long, give or take. And out here is the lumen of the intestinal, center. And then here are 2 enterocytes, showing off their microvillus brush border. A prettier way to look at this is with SEM, and we've been working with Matt Tiska and Evan Kristofiak at Vanderbilt to do some of these SEMs and kind of get a sense of what the brush border looks like here. And the more I work on brush border, the more you kind of see it everywhere in life. So I'm on the floor playing with my kids, and I look at the carpet and I see, oh, that looks like a brush border. I get out of the shower and I grab a towel and I dry off, and I look at the towel, and it, there's like a form follows function, I guess. All this absorptive surface of these little loops of cotton, I guess, that make a brush border. But here's about, I don't know, 24 cells on a villus. You can see some goblet cells poking out here, and kind of hexagons of enterocytes. And it's just this beautiful carpet of microvilli. And if you zoom in on a fractured epithelial cell with the inside of the cell here and the lumen up top, you can see the microvilli are just beautifully symmetrical, all kind of same height, all neatly stacking up next to each other, like tin soldiers or something. There's just a beautiful, beautiful structure there. So we know there's about 200 genes annotated in the gene ontology knowledge bases that localize, that their products localize to the brush border or the microvilli. And if you break down the genes that fall into these categories, you can kind of put them into 4 different categories. So first of all, there's an actin core. So a number of actin filaments all get bundled together by actin bundling proteins in blue, linking these actin filaments in green here. Kind of anchoring them to the membrane are a group of proteins that are in the membrane and also linked to the actin filaments. At the tips of the microvilli, brought up by motor proteins, are intermicrovillar adhesion proteins. So they actually kind of span the extracellular space between microvilli and kind of help them all stand up next to each other. And then finally, the business end of the brush border is a body of transporters that carry out the absorption and terminal digestion of the nutrients in our diet and suck them into the microvillus brush border. And so we'll talk a little bit about how all these proteins are regulated. And we're trying to learn more about how these are assembled. The TISCA lab's done a lot of great work, and this is a little schematic from their lab, describing that in the crypt, the brush border isn't really assembled the way it looks up on the villus. These microvilli protrusions are there in a rudimentary state, but they're kind of different lengths and sparse density, all rooted in an actin terminal web at the apical surface of the cell. And as the cells undergo differentiation and march up from the crypt onto the villus, they eventually will arrive at the structure that I mentioned to you earlier. So I'm by no means a cell biologist. We got into this because we've been very interested in gene regulatory programs governing intestinal cell function. And so we've long been interested in this crypt to villus transition as almost more as a process of developmental biology. How do you have stem and progenitor cells down in the crypts that are migrating up and literally hours later taking on a postmitotic differentiation program? How has that all worked out at the gene regulatory level? So we've done a lot of work, like Shahzad has, profiling epigenomic changes with things like TAC-seq or histone modifications, transcription factor binding profiling, to try to understand this gene regulatory switch. And just to kind of give you a sense of the dramatic changes that occur during this transition, here's results from a RNA-seq experiment where we collected either crypts or villi, extracted the RNA, and then compared the 2 transcriptional profiles between these compartments. Any given cell, you'll typically find about 10 to 12,000 of our 24,000 genes are expressed in any given cell. And about 1/3 of those expressed genes are dynamic across this crypt to villus transition. So when you run the gene expression analysis, you'll find about 2,000 genes are enriched in the crypt, and a separate 2,000 genes are enriched in the villus. And so, this tells us there's a lot of gene regulatory activity going on. And anytime we did, these kind of epigenomic screens, and I know Scott's on and Adam's on, they've done this kind of stuff as well. And you look at the sequences, at these enhancers that we think are dynamic between the crypt and the villus. We can informatically look for what transcription factors are binding and potentially regulating changes in gene expression. And HNF4 motifs are always right at the top of our lists. And so going back to work with, when I was in Ramesh Devdasani's lab, we really felt like, oh, HNF4 has got to be the key player. And so we and others have knocked out HNF4 alpha in the intestine, and There are some interesting phenotypes if you let the mice age or you perturb the system and with disease models. But at face value, when you look at the mice, they're fine. They live a pretty long and healthy life. They're fertile, et cetera. It was a little disappointing, kind of not congruent with the epigenomic stuff that we were seeing. But we know there's a paralog of HNF4 expressed in the intestine, HNF4 gamma. They're very similar in sequence and structure. And we suspected gene redundancy was at play. And so we went ahead and made a compound mutant to inactivate both HNF4 paralogs to maybe reveal some of the functions of these transcription factors in a deeper way. And that's where Lei Chen came to our lab. She's been in the lab for about 5 years now. And unfortunately for us, she's leaving to start her own lab at Southeast University in her hometown of Nanjing this summer. But she's done a great body of work on HNF4 transcription factors. And I'll share a little bit of that with you today. So what Lei noticed, and others have reported as well, when you inactivate HNF4 alpha or HNF4 gamma in the intestine on their own, the mice are pretty fine. They maintain body weight just like a wild type animal would. It's when you remove both of those HNF4 transcription factors in the intestine that the animals pretty rapidly lose weight. They don't survive more than about 5 days after losing HNF4 transcription factors. The intestines swell with kind of a fluid-filled lumen. And they have a number of phenotypes. And a lot of this is published now, so I'm going to briefly recap some of these, and then I'm going to get back to the brush border and focus on the brush border gene regulation for the remaining of the seminar. So we'll start off at the intestinal stem cells at the bottom of the crypt, specifically the crypt-based columnar stem cells that make LGR5 and OLFM4. If we look at these stem cells in the wild type or alpha knockout or gamma knockout, we don't really see much of a phenotype. But if we inactivate both HNF4 factors, by about 3 days, we start to lose OLFM4 immunoreactivity, LGR5 GFP expression, and by day 4, we really can't see these cells anymore. So I'll summarize a lot of work that Lei did here. But what we think is going on in the stem cells in the HNF4 mutants is that they fail to renew. We think HNF4 helps promote a metabolism in the stem cells that is based on oxidation of fatty acids. A number of groups have recently shown that conditions of high fatty acid concentrations in the body, either after fasting or in mice on a high-fat diet, lead to increased LGR5 stem cell numbers. We see a lot of HNF4 binding to fatty acid oxidation genes in ChIP-seq studies. A whole battery of enzymes that promote oxidation of fatty acids in the mitochondria that feed the TCA cycle go down in the HNF4 mutant. Conversely, genes that promote elongation of fatty acids or storage of fat into cytoplasmic lipid droplets are elevated in HNF4 knockout. And if we trace molecules like palmitate isotopically with metabolomics assays in the HNF4 knockout, instead of being oxidized and contributing to TCA components, in our HNF4 knockouts, we see these fatty acids are shunted towards either the elongated components of cell membranes or stored into lipid droplets. And we think that's part of the reason why we lose stem cells in the HNF4 mutant. Another phenotype that Lei noticed, and probably the most obvious one, is that when we knock out the HNF4 transcription factors, we see the cells want to kind of stay in this crypt transit amplifying cell-like state. So Ki67 marks the proliferating cells in the crypt in a wild type animal. You're probably all familiar with these structures already. And then the majority of a duodenal epithelium is villous, these really long villus, mostly with enterocytes and these brush borders. In the HNF4 knockout, instead of differentiating into these villi-like structures, the crypts just seem to keep getting longer and longer. There's more Ki67 positive cells. If you do whole amounts of crypts, you can see the elongated crypts in the mutant compared to the wild type. And so again, Lei did a lot of ChIP-seq studies and came up with the hypothesis that HNF4 can bind to members of the BMP SMAD transcription factor family and pathway. This pathway can also stimulate expression of HNF4 transcription factors. And then these two transcription factors can together bind to a battery of enterocyte genes. And so we think that these transcription factors are stabilizing each other's expression, kind of locking in an enterocyte identity, and then driving a battery of genes required for differentiation. And just to kind of give you an idea of the scope of the gene regulatory program under control of HNF4, I mentioned before there's about 2,000 genes that are elevated in the villus compared to the crypt. 70% of these genes are downregulated in the HNF4 double mutant. And in our ChIP-seq, about 60% of these are directly bound by HNF4. So that's a large portion of the enterocyte transcriptome that we think is under direct regulation of HNF4. And that's why we see these strong phenotypes and compromised function in the HNF4 double mutants. Okay, so that's kind of the background I have for HNF4 and the brush border. And now I'll launch into kind of what we think we've learned about HNF4 control of brush border gene regulation. If I'm losing anyone wants to ask a question right now, please feel free to interrupt.
This is great, Mike. Usually we end up asking questions at the end, so don't worry about it.
Okay.
You're right now.
All right, so now we'll get into specifically HNF4 control of brush border gene regulation. So here again are some of these nice SEMs from Matt Tiska. Again, this control, I think I showed a similar image earlier. In the HNF4 double mutant enterocytes, here's I think a couple enterocytes shown here. You can see that the microvilli are no longer kind of this uniform lockstep. They become this kind of eclectic mix of rebels. Some are tall and skinny, some are shorter and fatter, and they're less sparsely populating the apical surface of the enterocytes. If we look at a top-down view, you can see this nice carpet of brush border microvilli in the control, and in the double mutant, again, it becomes this kind of disorganized mix. So we can look at brush border gene products in our double mutant. First of all, when we trigger the double mutant, we can monitor HNF4 alpha loss by Western blot over time. We see about by day 3 after treatment, there's really not much HNF4 protein detectable. Villain is a actin cross-linking protein that localizes to the brush border. And by about 4 days after induction of the knockout, we no longer see villain protein. So that's strongly dependent on HNF4. Actin obviously is at the heart of the microvilli, the bundling that serve as the core of each microvillus. Actin protein levels stay pretty consistent. So we don't think HNF4 is directly compromising actin function. We can look in situ by IHC, actin or villain, and see them localized to the apical surface of enterocytes in brown here, and that their levels at the apical surface is quite reduced in the HNF4 double knockout. Similarly, alkaline phosphatase activity, which is the enzyme localized to the brush border membranes, that's also severely compromised in the HNF4 double knockout. Here's TEMs again showing the disrupted brush border at the apical surface of enterocytes in the mutant. This is from Nangao and Ed Bonder at Rutgers Newark. And just if we kind of break down the roughly 200 genes that we know contribute products to the brush border, about half of those go down in the HNF4 double knockout. And about 40% of all the brush border genes also have direct HNF4 binding by ChIP-seq. So again, we think it's a substantial portion of brush border genes are under direct control of these transcription factors. And I'll tell you a little bit more about our evidence for that in just a bit. So we have a lot of evidence in place that HNF4 is controlling the brush border gene expression program in the intestine, but the intestine is not, as much as my world revolves around the intestine, to the point where people give me plush versions of the intestine. We try to branch out a little bit sometimes, and other organs have brush border epithelium, transport epithelium, such as the kidney proximal tubules, or reproductive tissues like the placental labyrinth, or in the mouse, the mouse yolk sac actually has a villus epithelium. We wanted to look there to see if the brush border is regulated in a similar capacity as it is in the intestine. So we went back to these about 200 brush border genes, and we looked by RNA-seq at the kind of makeup of brush border gene expression in the yolk sac or in the kidney. We took all the genes that are expressed at FPKM over 1, considered those expressed, and then asked, are the same genes expressed? And the vast majority are found in the yolk sac, intestine, and the kidney. Many of the ones that are unique to the kidney versus the intestine might be like a paralog, like MYO1A, I think, is in the intestine, MYO1C is in the kidney. Sometimes it's a transporter, like a urate transporter is present in the kidney, whereas another transporter for maybe like a cholesterol transporter, like the Niemann-Pick complex gene, is specific to the intestine and not in the kidney. But the vast majority of these genes that localize to the brush border are common between these tissues. So that begs the question of how are these genes regulated in the diverse tissues? You could imagine that it could be diverse enhancers are regulating the same genes in different tissues, or the same enhancers could be regulating the same genes via the similar program in these different tissues. So to get a sense of, and answer that question, we wanted to collect all brush border enhancers. And to do that, we took advantage of DNA-seq data from the intestine and kidney. DNA-seq chromatin accessibility sites are typical markers of enhancers. And so we took annotated transcriptional start sites from brush border genes in the intestine and the kidney. We reached out 50 kilobases in each direction and collected all the regions that had a DNA-seq site. So now we had this collection of enhancers from intestine and kidney brush border genes. And we wanted to know what are the transcription factors that might be binding to these enhancers. We did that, asked that question using Homer motif analysis. Now we had a list of transcription factor candidates that could bind to these enhancers. And we wanted to check whether or not these transcription factors are expressed in these tissues based on RNA-seq data. At the end of all of this, we found a couple of things. One, we found that the enhancers that are open near brush border genes in the intestine are more often than not open near brush border genes in the kidney, suggesting the same gene regulatory process could be at play. We also found that in the motif analysis, some of the same transcription factor binding motifs were the most enriched in both tissues. The top motif we found belongs to CTCF, which isn't a transcriptional regulator. You're probably familiar that CTCF helps to kind of control the three-dimensional architecture of the genome into these topological associated domains. So a lot of the hits that we found are actually CTCF regions of the genome. But the second most abundant category of factor we found enriched is the motif for HNF4 transcription factors. So we were excited about the idea that HNF4 could control brush border in the same way across all these different tissues. Here's just a look at HNF4 expression in red in the nuclei of villi cells in the intestine or proximal tubule cells in the kidney. So we compared HNF4 ChIP-seq in the kidney and HNF4 ChIP-seq in the intestine using an analysis called diffbind, and found that there's a remarkably similar binding profile in these two different tissues. And so, you know, the potential for a similar gene regulatory program in the intestine, kidney, and yolk sac was there. But functionally, would HNF4 be important in these different tissues? If we knock out HNF4, does the brush border gene expression program decrease in these different tissues? Is there some kind of physiological consequence associated with the brush border kind of manifested upon knockout of HNF4 in these tissues? So in the intestine, we had to knock out HNF4 alpha and gamma to see a phenotype. In the kidney and yolk sac, we don't detect any HNF4 gamma expression. So we could use the ubiquitin CRE to inactivate just HNF4 alpha, and we think we would lose HNF4 activity. So we'll start by looking at some gene regulatory consequences of HNF4 loss in the kidney and intestine. First of all, the ChIP-seq, if we see a HNF4 binding peak in the intestine, we often see a HNF4 binding peak in the kidney as well. Again, consistent with that diffbind analysis. If we look at the RNA-seq data of this gene MYO7B, which is a motor protein that helps localize intermicrovilli proteins to the tip of the microvilli, each of these kind of bundles of sequencing reads represents an exon of the MYO7B locus. And the controls gray, and in red or blue is the HNF4 knockout in the intestine or kidney. Hopefully you can see that the levels of sequencing reads are going down in the knockouts, suggesting MYO7B levels are going down. Espin is another actin microvilli bundling protein, where HNF4 binds many of the same peaks in intestine and kidney, and the levels of espin go down in the HNF4 knockouts of both tissues. A little bit about HNF4 binding and enhancers. So here's an example where we see HNF4 binding about 200 kilobases away from the espin promoter in the mouse intestine. To get a sense of whether HNF4 is really working from this enhancer to regulate the espin promoter to control its gene expression, we did an assay called HiChIP, where we can ask whether or not two regions of the genome are close together in three-dimensional space. Briefly, the way this works, I won't get into the weeds, but I'm happy to talk about it to anyone if they're interested later on, is you cross-link the genome in its three-dimensional conformations, hoping to capture enhancer-promoter contacts. After you cross-link, you kind of permeabilize and send in a 4-base cutter to digest up the genome into smaller fragments. And then you put in a ligase to try to reattach the cuts that you just made. What you're looking for is an enhancer DNA region ligating to a promoter DNA region. The only way that would happen is if they were in proximity to each other. There's some other tricks in here, like you ligate in biotin to enrich ligation events. You also immunoprecipitate a promoter chromatin mark, histone 3 lysine 4 trimethylation, such that when you go to do the sequencing, you have mostly promoter regions of the genome pulled down. And then when you do paired end sequencing, you hope to capture a read that starts with enhancer DNA and then crosses over to a promoter DNA fragment. And then you can count up those non-native ligation events as evidence for a looping event. If you have a lot of reads that are between an enhancer region and a promoter region, that suggests a strong chromatin looping event. If there's fewer reads, that suggests a infrequent enhancer promoter contact. So this is a semi-quantitative approach to look at enhancer promoter interactions. Basically, what we're hoping to see is enhancers near brush border genes are near in proximity to promoters, and they correlate with expression of these brush border genes. In the HNF4 alpha gamma knockout, we wonder whether or not these contacts are less frequent. That would tell us that HNF4 is helping to facilitate enhancer promoter communication and stimulate gene expression of brush border gene. We found a lot of looping events in the villus and crypt. The looping events change in the HNF4 knockout. I won't go into all the details here, but I'll get back to our specific example. These are all published if anyone wants to get into more details on that. But if we go back to our region of the genome bound by HNF4, about 200 kilobases upstream of the espin promoter, we do detect ligation events between these regions of the genome in the wild type. And in the knockout, we count fewer ligation events, suggesting that the looping is occurring less frequently. We also see these events happening between downstream binding sites of HNF4 and the espin promoter. And then we can try to use orthogonal data sets, such as measuring chromatin modifications in the wild type or in the HNF4 knockout, and see if all these things line up to suggest HNF4 is controlling expression of espin or other brush border genes. So histone 3 lysine 27 acetylation is associated with enhancer chromatin. And you can see this HNF4 bound region has this mark. You can see other regions bound by HNF4 have this mark enriched. And when we knock out HNF4, we see this mark is lost at HNF4 binding sites in the genome, but not really affected in non-HNF4 binding regions of the genome, which serves as a nice internal control. And at the end of all this, what kind of really matters is that espin gene expression levels are going down quite dramatically in the HNF4 knockout compared to the wild type. So this is the kind of analysis we've done to try to implicate HNF4 in regulating the brush border gene expression program. But does any of this have functional consequences in these transport epithelium, where HNF4 again is expressed in the kidney proximal tubule cells, in the yolk sac epithelium, and in the intestinal epithelium? Just a little more detail on the proximal tubule cells marked by ABCG2, a urate transporter in red. You see the yellow HNF4 nuclei near these cells. The distal tubules of the kidney express a sodium chloride cotransporter. HNF4 is not associated with these cells, and these cells coincidentally don't make a brush border. So we knock out HNF4 in the kidney, either in the neonatal stages or in the adult. We see very similar consequences. We did RNA-seq in the kidney after knocking them out. We collect urine and we collect plasma from these animals. And we look for changes in the ultrastructure of the proximal tubules as well. So now we're looking at one proximal tubule by transmission EM. And in the lumen of the proximal tubule, you'll see it's really filled up with these long microvilli. It's a pretty cool ultrastructure in this tissue. In the HNF4 knockout, we can start to see an open lumen, and the microvilli are much shorter. Hope you can appreciate that there. We can stain for markers of brush border. I think I skipped it. Yeah, there we go. Like alkaline phosphatase activity. And it hits this is the cortex of a kidney shown here. And you can see alkaline phosphatase in a pattern consistent with proximal tubules. And HNF4 knockout, we lose that enzymatic activity. HNF4 immunoreactivity is also in the proximal tubule cells. We lose that in the kidneys. And we've done a number of other histological analyses to show that this brush border gene expression program is really compromised at the protein level as well. And then finally, at the RNA-seq level, if we use gene set enrichment analysis, we basically arrange the HNF4 RNA-seq into genes that go up and HNF4 knockout on the left, genes that go down and the HNF4 knockout on the right, and ask where the brush border genes lie in this continuum. We see that they're asymmetrically distributed to go down in the HNF4 knockout, just like we saw in the intestine. And this is deemed statistically significant. Finally, some physiological metrics of a compromised brush border. The brush border reuptakes whatever you don't want to filter out of the blood as you're making urine. Glucose is one of the big ones. And so you see glucose is up in the urine of the HNF4 alpha knockouts. The plasma becomes more acidic, probably because the kidney's not taking out bicarbonate properly. Ions like potassium and calcium in the plasma are going down, as well as glucose going down in the plasma. These are all phenotypes consistent with what's seen in Fanconi patients. And many Fanconi patients actually have genetic mutations in HNF4 alpha. So we think these models might be actually interesting and applicable to this disease. Okay, so finally, the last tissue I'll talk about is the weirdest one for us. I didn't know the yolk sac of the mouse had a brush border until we started this project and started looking for brush borders. But it turns out that if you look at a murine placenta, if you section through it and you do an HNF4 immunostain, and what you'll see is, this is the placenta tissue here, this kind of disc, which is quite large. And then this really thin yolk sac membrane kind of projects off of it and encases the embryo. And so the embryo would normally be kind of in this area, and outside the embryo in the uterine lumen would be up here. And especially at the yolk sac proximal to the placenta, you see all these finger-like projections that are quite reminiscent of intestinal villi, and they're strongly immunoreactive for HNF4. I hope you can appreciate that. Going back into the 80s, SEMs were done showing the surface of the yolk sac epithelium, and they're actually lined with a brush border microvilli, which is pretty cool. And so we wanted to, again, knock out HNF4 in the yolk sac, and then look at whether or not brush border, protein expression was compromised. ABCG2 is a urate transporter, again, that marks the brush border in the intestine, the kidney, and the mouse yolk sac. And you can see it lining here, the apical surface of these cells. In the HNF4 alpha knockout, the levels are quite reduced. Similarly, if we look at beta actin, which total levels of actin don't change in the HNF4 knockout, but they no longer are concentrated to the apical surface of these epithelial cells. Instead, they're more towards the lateral surface of these epithelial cells. And then finally, vilin, again, localized to the apical surface where there's a brush border, is significantly down in the HNF4 alpha knockout. The function of these villi is a little bit, I'd say, poorly explored. Why is there this transport epithelium on the murine yolk sac? So we took a little bit of an unbiased approach to just see what could be going on by collecting amniotic fluid from all the embryos in a litter, where kind of half the embryos were HNF4 alpha knockout because they had the Cre. The other half of the litter didn't have the Cre, so they were HNF4 wild type, but they all got exposed to the same tamoxifen. They're all in the same maternal environment and everything. We wanted to measure if there were any changes in the metabolites. We saw a couple of things quite strikingly. One was that amino acid levels were down in the mutants amniotic fluid, and a number of amino acid transporters are expressed at the brush border of the yolk sac epithelium. We also saw that uric acid levels were higher in the knockout amniotic fluids compared to the wild types. And this makes sense because urate transporters ABCC4 and ABCG2 are quite dramatically downregulated in yolk sac from the controls versus the HNF4 mutants. So we're not sure. The embryos are smaller in the HNF4 alpha knockout using the UBC Cre. Maybe it has something to do with the yolk sac function. I can't say that confidently because this system, we're also knocking out HNF4 alpha in the developing intestine, which appears normal because HNF4 gamma is present, but also HNF4 is expressed in the liver, in the pancreas, and in the kidneys, as I mentioned earlier. And while we're not sure about the function of these tissues in utero compared to their importance in the adult, certainly I'm not anyway, we do see this phenotype and we think it's rather interesting. We have to develop a Cre specific to this yolk sac villi to really parse this question. But at least at the gene regulatory level, again, if we look at the brush border gene signature in the yolk sac, we see again it's downregulated in the HNF4 alpha knockout compared to the control. So taken together, we think HNF4 controls a brush border program that's largely conserved between these three tissues and important for the function in each of these transport epithelium. Okay, so I'll go on briefly to just discuss where we'd like to take this work. One area of interest that we found is when you, again, knock out HNF4, we don't see actin in the brush border anymore. Again, actin's like the core of those microvilli. And it's not going down. The total actin levels aren't changing, but what we see are stress fibers forming. And obviously this can change mechanosensing in the cell. This can change potentially tight junction formation, barrier integrity, a number of functions that maybe will have implication for IBD or other compromised epithelial functions. So trying to understand how that's regulated and if that's an important part of the HNF4 phenotype, I think will be an interesting area to research. HNF4, as much as a prophet of HNF4 as I'm trying to be, it's not the only transcription factor controlling these gene regulatory processes. We've shown previously that knockout of the transcription factor CDX2 compromises brush border as well. Really no transcription factor works on their own, but they bind in complexes, maybe 4 or 5 different transcription factors at an enhancer. So HNF4 could work with CDX2 to help promote brush border in the intestine, but in the kidney, we don't see CDX2 expression. So is there another transcription factor there partnering with CDX2 to drive the brush border gene program? That's an area that we think is interesting. One of the coolest areas I'd like to explore is asking whether or not the brush border is somehow, we know it's very dynamic. Matt Tiska has some beautiful live imaging of the brush border. It's very dynamic. It's not just standing there kind of fixed like statue. It's dynamic in response to different signals. It can kind of flay its microvilli out to increase surface area. Different pathogen infections can change brush border structure. And there's a stoichiometry problem here where if you're localizing at least 200 gene products to the brush border, how are they assembled with the right numbers to achieve a brush border that's typical of the intestine or kidney? So we think that there's kind of communication between the brush border and the nucleus. One hypothesis is that a protein like filament A, which can localize to the actin cortical web, could be sensing the brush border integrity. It's proteolytically processed and can send a short fragment to the nucleus. And we and others have seen that HNF4 can physically interact with filament A in co-IP studies. So we're wondering if that's a component of gene regulation between the brush border and HNF4. And then briefly, I mentioned pathogenic strains of E. coli, Vibrio, other pathogens can bind to the epithelial surface and trigger a restructuring of the brush border epithelium, the brush border structures. And if any of that has kind of a gene regulatory component, we think maybe HNF4 is involved in that. In support of that idea, if you look at mouse epithelium that is infected with EHEC, you see a substantial downregulation of the HNF4 gene expression program. So how that's all communicated, I think, is going to be very interesting and potentially important in developing ways to handle these types of infections. So that's it on brush border. I'll just briefly go over a couple other projects in the Verzi lab. So we're interested, again, generally in gene regulation, including epigenetic regulators. And Win, a graduate student, is working on histone acetyltransferases, CAT2A and CAT2B. When Win knocks them out, we completely lose H3K9 acetylation and see some cool phenotypes, including activation of interferon signaling. And so Win's actively trying to solve that puzzle and try to understand why that happens. Kieran Vimuri in the group is looking at HNF4 and partner transcription factors in diet-induced obesity by combining some physiological models as well as epigenomic regulatory processes. We're also interested in colon cancer and specifically the initiation of colon cancer, how stem cells respond to an oncogenic mutation, how the stroma responds to a change in the epithelium, how it senses it and changes its kind of niche supporting profile. And Oscar Payon Cardenas and Kevin Tong are working on these projects. And then finally, we've been doing some study on vitamin D induced gene regulation, specifically asking the question in partnership with Sylvia Christakos and Jim Fleet. How does vitamin D control calcium uptake in the duodenum versus in the colon? Is it through similar regulatory pathways? Is it through different? And some of the other functions of vitamin D, are they regulated in similar processes in different regions of the gut? Joe Herr, a technician in the group, has worked alongside with a team of undergraduates, Dennis Aldea, Rohit Aita, Soheb Hassan, and Evan Cohen on these projects. So that's it. I'm blessed with a really great group, mix of undergraduates, Tech's graduate students and postdocs, and child labor laws that help us to push these questions and try to solve some of these puzzles. We're also blessed with a number of great collaborators. I mentioned many of them along the way. Jason Spence is also a fantastic collaborator at the University of Michigan, working on some stem cell projects. I mentioned my postdoc advisor, Ramesh Devdasani, who we still collaborate with on some projects. Collaborators in the Department of Genetics at Rutgers, Jin Jing, Chris Ellison, and Amrik Sahota. Steve Brandt on IBD gene regulation is at Rutgers Robert Wood Johnson Medical School. And then the metabolomics work was done in collaboration with Xiaoyang Su at the Cancer Institute in New Jersey. So I'll stop there and be happy to take any questions. Thanks so much again for the invite. And this was a lot of fun to get to spend some time with you guys.
Well, that was fantastic, Mike. Thank you for your presentation. The questions will start rolling in a bit. If you guys have questions, you can let me know and I'll call on you. But I'll start with Kendall. Kendall, you just want to ask your question directly?
Yeah, sure. Really cool work. Thanks for sharing. I was particularly taken as a cell biologist. I was taken with the kind of redistribution of actin away from the apical surface of the brush border and the yolk sac cells. And just wondered if any of the kind of classic apical polarity genes popped up as altered by loss of HNF4 in your RNA-seq data.
Yes, so that's a great question. And I was recently talking with Jim Goldenring at Vanderbilt, and we were looking at some of the the RABs and whether or not they're regulated at the transcriptional level. At first glance, I would say it's not as striking with my limited knowledge, but I should probably share that data with someone like you that can really go into it and know what all those, how all those different genes function, and how much of the HNF4 phenotype could be explained by trafficking problems more than, expression of the structural components of the brush border. I think that's a great question, Kendall.
Thanks.
Yep. Yeah, I don't know if it's just, you depolymerize the actin and then it needs somewhere to go. I don't know if these cells are stressed and that's why they're forming the stress fibers, or if it's other mechanisms like the one you proposed. It's all a new area for us. I think it's a great question.
Yeah. It's really hard to tease apart, right? Because all of it's kind of connected. And with it, I have no idea if anybody's looked at the role of, PAR complex or crumbs or some of these other kind of well-known polarity systems and even just intestinal brush border. I have no idea, but it'd be kind of fun to look at.
Yeah, for sure. We've been looking at partner transcription factors of HNF4, hoping that, if we knock out a specific partner, we'll find one that's, the partner that helps control brush border. This is the partner that helps control lipid metabolism. This is the partner that helps control, maybe some other aspect of enterocyte physiology. And so far, it hasn't been that crystal clear. There's a lot of, crossover, I think, between a lot of these functions.
Hey, Verzi, I'm Matt Tillman, and I actually work in John Rawls' lab and work with HNF4 alpha. So I'm very interested in your talk and really enjoyed it. So I was interested, we've been very interested in understanding, the utility of alpha and gamma in the intestine. And I found it interesting how, you have these other tissues like the Yolk sac and the kidney where you're seeing alpha alone is mediating these effects. And so is it truly just full compensation, you think, in the intestine, or what do you think the purpose is of having gamma in the intestine? So I'm just curious of your current thoughts about that.
Yeah, I think it's really interesting. Why does gamma pop up in the intestine and not in these other tissues? One thing that we do notice as a difference between these tissues, or between these paralogs, I guess, is gamma seems to be more restricted to the villus. Other investigators have delved into alpha functions in promoting proliferation in the crypts. There's different isoforms of alpha. One's more expressed in the crypt and the other in the villus. Francis Ladek has done some nice work in this area. So those could be some differences to kind of help HNF4 take on those unique functions. Maybe that's one of the reasons that this process evolved. I know from talking to John and the zebrafish, there's even more paralogs. I don't know if that's an accident of, the genome duplication in that organism, or if there's also some different functions. It'd be interesting to see, what you guys find, in that model as well, to try to help pick apart, some of the potentially different functions. On high-fat diet, we see different phenotypes. We see gammas gain weight on high-fat diet and alphas don't. So there's something different there. And I think exploring, I know you guys do a lot of nice work looking at the intersection of diet, microbiome, HNF4. So, I think there's certainly reasons that you have paralogs and they evolve. But in terms of me telling you specifically what they are, we haven't made a lot of progress on that yet.
Yeah, thank you.
Yeah.
Can I jump in? Hi, Mike. That was great. Very, very clear. Really nice to see what y'all been up to. So I guess this is really building off of both Kendall and Matt's question, but well, there's really two questions, I guess. So if you think that the HNF4s are, really driving a brush border program, have you, when did brush border as a eukaryotic cell invention, when did it arise? And is that matched up with HNF4 emergence on the phylogenetic tree?
Yeah, we tried to tackle this by looking at estuarine crabs. So they kind of make a brush border to osmoregulate dynamically as the tides come in and out. And so we were trying to get tools to look at HNF4 in that system because I thought that would be so cool. But we just couldn't get the tools and the systems up and running. But that was one of our original ideas, you know, to see is HNF4 regulator across kingdoms of brush border. And it just never manifested. I think it would be really fun to look back and to do some Evo-Devo stuff with that. And it wasn't something that we could kind of pull together. And so instead for this project, we went into different tissues within the mouse to see, you know, are these different brush border all under control of HNF4? The cells in the inner ear make a cool structure for, you know, auditory sensation. The vibration of the microvilli is what helps produce the sensation of sound, of hearing. HNF4 is not in that tissue. So there are other ways to make brush border. That's not a transport epithelium. So maybe that's a component of what HNF4 does. But yeah, I think it's cool. I mean, you guys are more, you know, diverse in the species you study. And if you or someone you know wants to pick away at that, I think it'd be really fun.
Seems like Drosophila would be a good place to start too, just a single gene.
Yeah, I don't know about, you know, what the different transport epithelia there are, but even just looking in the intestine, right?
Yeah.
That'd be cool.
And then I guess the other question is, you know, bringing it all the way back to mitochondria. I mean, how, you know, you make a really compelling case about, you know, the HNF4 is having an important role in brush border in multiple tissues. And then, you know, yours and others' work kind of suggests the role of mitochondrial function too. And so how are you kind of reconciling those things in your head? You know, I mean, on the surface, they seem unrelated, right? But how do you think about those two roles? Do you see them as related? Do you see them as, you know, just, I don't know, you know, having evolved independently? Like, how do you think about that?
Yeah, you know, and it's fun to think about all the kind of evolution of all these things. It's fun to talk to you about that. Hopefully we can get together, you know, sometime soon and get all philosophical. You know, HNF4 has important lipid regulation roles in the liver. You know, if you go back to organisms like the worm, right? The worm guy is probably part liver, part enterocyte. And so, you know, is HNF4 generally involved in the identity of these cells? And all these things fall under the umbrella of HNF4 dependent transcription. I think it's argued that HNF4 is kind of the closest thing to the founding member of nuclear receptor transcription factors. So yeah, I think, you know, a lot of the gene regulation could have started under HNF4's purview. And then a lot of the diversification of all these cellular functions kept HNF4's input as one of the initiating regulators to open up these regions of the genome. We have shown if we knock out HNF4 in the developing gut, enhancers associated with mature enterocyte function aren't able to open. So we think it might lay the groundwork for what comes later in development. And others have shown in the liver that, again, HNF4 is required as, you know, I don't know if we use the pioneer factor term, that's become a little bit controversial, I guess, but is HNF4 required to open up the enhancers that lay the groundwork for multiple cellular functions to occur, including beta oxidation, fatty acids, assembling a brush border, et cetera? That would be my guess, but it's all speculation.
That's great, thanks.
Anybody else? All right, very cool. Mike, one last question, then we'll let you go. I have to ask this because the role of short-chain fatty acids in the colon, you didn't really talk much about it. I understand the brush border link, but I'm wondering if you've managed to see any form of pathology in the colon in your knockouts and double knockouts, even at a histological level or under stress.
Yeah. we put HNF4 alpha mice on DSS, and we see that they do poorly. That's consistent with HNF4 being associated strongly with human IBD genetics. Other labs have done that as well. John's group has shown HNF4 alpha mice are prone to inflammatory bouts. Most of the phenotypes that we've described in the double mutant are most severe in the duodenum, and then they get less severe as you move down towards the colon. We think that tracks basically with HNF4 gamma expression, which is higher in the duodenum and gets weaker and weaker as you go towards the colon. And so we think the redundancy phenotype is more focused towards the proximal tissue. Why the colon, is less dependent on HNF4 factors is a good question. If we think about the functions that we've attributed most strongly to HNF4, which is nutrient absorption and lipid metabolism, those I guess are more proximal functions of the intestine. Whereas in the colon, I guess the phenotypes there maybe are more associated with barrier integrity and not as HNF4 dependent.
Right.
But that's not to say there's no role. It's just not as acute as what we get in the proximal duodenum.
Excellent. Well, thank you so much.
Yeah.
This is a wonderful discussion, and
yeah, it was a lot of fun.
Yeah. Thanks everybody for attending. Mike, have a great evening.
Thanks, you too.
And hopefully we'll see each other in person one day.